Swayam Prabha. Digital India. Educated India.
Hello all, welcome back to our sessions on precision oncology. Last class we have got a very basic introduction of cancer immunology. We have really went into the history of the cancer immunology, we've learned about immunosurveillance, we have understood what colis toxins are, and we got exactly the introduction of the innate and the adaptive immune responses. Here we will continue our work, our sessions on cancer immunotherapy. Just coming back, to the basics. First, you have your adaptive immunity and your innate immune mechanisms. So, the major cells of your adaptive immune response are your B and T cells with specific antigen receptors. Here, do you see them? These T cells are differentiated, are divided into two populations by differentiation in the expression of you have your T helper cells and your CD8 cytotoxic T cells. This CD4 T helper cells are further subdivided into your Th1, IL2, IL5 which secrete and your Th2. This is all the regular basics. And then B lymphocytes also have a role in your adaptive immunity via recognition of your foreign antigen through various B cell receptors, and whereby a lot of your antibody production happens. Then you have your antigen presenting cells, which are APCs, which are essential for your very essential for your adaptive immunity, and especially these APCs are your first antennas for our cancer immunotherapies. These are essential for your adaptive immunity. They interact with your Th and the T helper and the cytotoxic T lymphocytes. This innate immunity response, which represents, this is like for your general, your regular antibody infections and regular viral infections. This is all of innate immune response happens. This is like an immediate, and this is by inborn innate mechanisms, and this usually occurs by occurs by the environmental pathogens or harmful any other harmful effects of the typical. Your macrophages produced inflammatory mediators here, what we can see here, such as your IL1, your IL6, whatever here, whatever we all see here, this is here. And then you have even your basophils and your eosinophils also, which form a part of your innate mechanism. This is with respect. Then you have very one crucial cells such as your dendritic cells, and then you have even the complement pathways as part of your innate cells. Then you have your this activated natural killer cells, which are which produce a number of cytokines such as your IFN alpha, your TNF alpha. Recollect in your basics of cancer biology, we were discussing about the cytokines. Then your GMCS, CSF, and then your very importantly your stimulating your cytokines, very important, like CCL1, CCL2, CCL3, 4, 5 and 8. These dendritic cells represent the central regulators of your specific immune responses. They will be presenting your antigens to the helper T cells and B. This is all the basics. Then now coming, what happens, we will introduce to the small terms like, so this is how exactly your T cell and then your antigen present. How do these two cells interact? You have very fantastic receptors usually, and these many of these ligands which are represented by your antigen presenting cells, they can bind to many multiple receptors, and they deliver stimulatory or inhibitory signals. These inhibitory signals are referred to as your immune checkpoints, which we will see in the next slides. Antagonistics of your stimulatory signals, or those which are antagonistic of your inhibitory signals, they will be resulting in a cascade of your amplification of your specific T cells. Usually activation of your T cells, as we have seen last time, requires two signals which are delivered by your antigen presenting cells. The first signal is antigen presented in the form of your peptides. We all know, right? All these peptides are processed by your major histocompatibility complex, MHC complex. This peptides is recognized by this T cell receptors. By the T cell receptors here, these peptides are recognized, and they are and this interaction provides specificity. It is a very specific T cell response. The second, apart from this particular regular signal, you even have a co-signaling stimulatory ligands on APCs. These co-stimulatory ligands will interact with the corresponding receptors on the T cell surface. Sometimes without a co-stimulatory interactions, the T cell will either die or they become energetic. How do cancer cells become tolerant to this immune system? What did we first discuss? We discussed that cancer cells have to evade the host immune response, that they have to be, now if they do not have to be recognized as foreign. They can become tolerant to this immune system by upregulating the expression of your immune checkpoint molecules like your program cell death. This is a very, very important ligand, PDL, program cell death molecule. So, here and then which leads to peripheral T cells by the time because of this prolonged expression of your PDL1, it leads to peripheral T cell exhaustion, what we have discussed last time, or loose surface antigen expression, which leads to immunological escape. Your immune checkpoint inhibitors, what do? They will bind to this. Suppose you have anti-PDL1, the monoclonal antibodies will bind to this particular anti-PDL1, and these inhibitory compounds currently they are used in pharmacological interventions using the three different ligands. We will be talking about this particular 3 different ligands. As I mentioned, this table will give you about what are the different co-inhibitory. This is on the antigen presenting cell and this is on the T cells. The T cells require your very much, as I said, your co-stimulatory signal for optimal proliferation, differentiation and survival, making co-stimulation necessary to produce immune response. This cancer the immunotherapy has received, it has received a lot of breakthrough only after they have established this particular co-stimulatory receptor ligand pairs, that is when they totally understand not only the ligand, even a co-stimulatory ligand receptor ligand pairs are also very, very important. For example, if at all you are able to block any of this co-stimulatory and co-inhibitory pathways, you are or maybe if you can optimize the use of this co-stimulatory and co-inhibitory pathways, it has a potential to increase the effective T cell mediated anti-tumor immune response. So, thereby you can increase the efficacy of your cancer immunotherapy. Cancer immune checkpoint molecules, they refer to a group of immune receptors that upon interaction with their ligands, they transmit an inhibitory signal to suppress your effector function. Very simple itself, there is nothing like. First we talked about what exactly are this particular, where exactly you have a ligand and then you need to have a co-stimulatory molecules, and what are these immune? This is a checkpoint molecule. Do these checkpoint molecules, are they important in the normal function? Yes, these important pathways, inhibitory pathways, they are very, very crucial for maintaining the self tolerance and regulating the intensity and duration of immune response in your peripheral tissues to minimize the tissue pathology, and the same pathways can be used for transfer to evade tumor immunity. So, isn't it wonderful that whatever is existing, these pathways are only being used for your cancer immunotherapies. Can we have a blockade of immune checkpoint? Yes, they can be blockade, and this blockade of these particular inhibitors, if you have inhibitors for your, as I said, anti-PDL1, they will be effective in a variety of tumors that are refractive to, for example, this particular immune checkpoint therapies. Sometimes the patient is not responding to a particular chemotherapy or radiation. Yes, anti-PDL1 monoclonal antibody along with your radiation therapy. This immune checkpoint inhibitors, yes, they are very well currently approved by your US FDA administration for the treatment of various cancer types and have significantly improved clinical outcomes and survival. Antigen presenting cells process and express antigens including your tumor antigens. Do you recollect we were talking about tumor specific antigens or the neoantigens in the last session? And so if at all, what a wonderful it is, like you have a specific antigen. On major, these are recognized on MHC complexes which are recognized by your receptors on T cells, and they stimulate a cascade either to kill the expressing the antigen via your CD8 effector cytotoxic T cells, or they will recruit your other components of the immune system via your CD4 plus helper T cells. This gives a complete again your overview. These are the different families. You have the BCD28 families, you have the TIM family, and then you have the immunoglobulin superfamily, nectin and nectin-like families, and the BTN and the BTN like families. So, you have different all this immune checkpoint molecules and the co-inhibitory receptors including your immune which cause your immune suppressive conditions in the TMV. They are all differently grouped. So, this slide will represent your overview of your several immune checkpoint inhibitors, co-inhibitory receptors causing immune conditions in your tumor microenvironment. You have the different families like such as your BCCD28 families, your TIM family, and then your immunoglobulin superfamily, then the nectin and nectin-like family, and the BTN and the BTN-like family. We see very how nicely all these different receptors are represented here. On the CTLs you have the T cells, you have the CTL4, then you have the PDL1, then you have the CD80, PD1, Vista, then CD60, BTLA. These are all coming. Please keep in mind your B7 CD28 families of co-inhibitory receptors. Please keep in mind this that B7 CD28. These are all very important co-inhibitory receptors because we will be looking in detail into them when we talk about our CAR-T therapies also. So, here this is like again your TIM families has your TIM3, and then here they will have the receptors such as your CEA, CAIMT and galactin. And then whatever here your immunoglobulin superfamilies has this particular receptors. You have different immune checkpoint molecules are expressed on T cells, and these whatever immune checkpoint molecules are expressed which are expressed on your T cells, they are shown along with their ligase expressed on your antigen presenting cells. Isn't it is on your antigen presenting cells or on the T cells or on tumor cells and or T cells which trigger a co-inhibitory signal molecule. Now, cancer evades the immune system. We all know this is all we have established, we have totally understand, it's a very established fact, and we totally understand yes for its survival. How does it? By use of regulatory cells, and then it uses anti-inflammatory cytokines, then it has decreased stimulatory receptors, and then defective antigen presentation, T cell tolerance and evading of your apoptosis. Now coming to your this is like a regular like you recollect we were discussing in the last class anti-tumor immunity, how exactly, isn't it a very fantastic, like tumor your T cells recognize tumor antigen which triggers T cell activation, and then suppose if everything it is presented by your MHC class molecule, the MHC class. T cells are always the preferred immune cells against targeting in cancer through their capacity to kill. They have a capacity to kill your malignant tumor cells upon recognition by the T cell receptors. These are your T cell receptors, and then which is presented only by your MHC molecule. This MHC molecules class 1 can be down regulated on tumor cells. Either these, isn't it like, so what happens if they can be down regulated in tumor cells and on CD8 T cells, they are, so obviously your CD8 T cells they cannot recognize, they will not be able to recognize tumor cells. Furthermore, you even have your loss of antigen variant on tumor cells which leads to tumor recognition by CD8 T cells. Then T cells are also, so this is all non-classical HLA molecules. T cells are not able to recognize your tumor cells because of your loss of the variant. Then another one is T cell activation is completely inhibited. So now how is this happening here? Here you can clearly see that T cell they are inhibited by the involvement of your inhibitory receptors and their corresponding ligands here. Then immunosuppressive cytokines are which will lead to suppression of your anti-tumor response are expressed. And then your regulatory T cells, your T regs, which are here, they will be they will suppress the T cell response to tumors by wherein your myeloid derived suppressor cells, they accumulate and suppress anti-tumor T cell response. This is in just by how the immune response is evaded by your T cells. Now we got introduced to the term TILs last class. Suppose there is a lot of TILs, presence of TILs, it requires a good prognosis for what are TILs? Tumor infiltrating lymphocytes. They represent a lymphocytic population. For example, as I told you before, always keep in mind, never a cancer cell is only cancer cell per se. This is a perfect diagram from biorender where you see your tumor cells, your cancer associated. You recollect in the last class, like we were talking about how a tumor TME in pancreatic cancer, how does it look like? It has a lot of lymphocytes which represents your CD4, then there are a lot of cytokines released, and there are many growth factors. These are your macrophages, then these are your tumor cells. Then you have many other different cells, that and these cells, they exert divert effects on the there's a lot, not only like what towards the tumor sustenance, and they have a lot of effect on the immune response. So subsequent to antigen stimulation by your antigen presenting cells, you have the activated CD4 T helper cells. These cells stimulate cytotoxic T lymphocytes by IL-2 secretion and cell-cell interaction through your co-stimulatory molecules, including your MHC class 2 molecules, the CD27 and CD134. Cancer cells, by producing CD18, they recruit your Tregs. Very important, as I mentioned in my last slide. Here you even have the cancer stem cells which have the stemness markers such as your CD24, CD44, CD47. Then you have the self-renewal pathways which are activated, such as your WNT, your beta-catenin, then you have the hedgehog, then you have the increase in your EMT, then all this will lead to increase in drug resistance, and then you have the tumor relapse and the tumor recurrence occurs. This is like, you have interaction with all your different even your non-cytotoxic CD8 T cells also lead to your cancer stem cells. So different, see, if you see all these cells, your natural killer cells, your dendritic cells, myeloid derived suppressor cells, then your tumor associated macrophage, the T lymphocyte, T regulatory cells, all these are which are parts. The same colors are represented in this particular the tumor. This is the tumor, the whole tumor mass, and here this is your blood vessel. As a result, this is what happens in your T lymphocyte. There is your increased in PDL1 expression, then decrease in CD4 and CD8 T cell proliferation, and therefore it is like or a decrease in anti-tumor response. So, and then what will it lead to? Again, it will lead to your cancer stem cell pathways which are all triggered. Then your regulatory cells, what will happen? There is increase in population expansion. So, there is an increase in immunosuppressor effective plasticity such as your T70 Treg balance. It is like, this is like supposing you have your TILs, then tumor infiltrating lymphocytes, which is always represent. How does this tumor infiltrating lymphocytes play a role? We have even introduced ourselves last time to another concept called immune exhaustion. What exactly we totally defined it, which and this immune exhaustion, it also contributes to immune dysfunction in cancer. What happens, the activation of your T cells is regulated by the balance of inhibitor, that is a checkpoint, and a stimulatory pathway, which we have already established, and this is very critical in your normal healthy cells to prevent your autoimmunity, that is self tolerance, and to protect normal tissues when the immune system is activated against a pathogen. During long term antigen stimulation, that is exactly what happens when you are cancer. Your effector function of CD8 D cells gradually decreases, and this phenomena is known as the exhaustion. The process of T cell differentiation into effector and memory C cells is altered, and this whole switch is happening towards a particular stage called exhaustion. In you have inhibitory receptors may act during both immune activation and ongoing immune response. Many during chronic inflammation also, these T cells, they become exhausted and they upregulate a wide range of non-redundant inhibitory receptors that limit their effectiveness. The whole purpose of your T cell effectiveness is totally lost when you have the increase in your receptors such as your PD1, CTLA4 and your T cell immunoglobulin. This interaction of the between inhibitory receptors and their ligands, it will reinvigorate intratumor CD8 T cells. Now, just a very brief overview how the receptors are associated with immune inhibition and dysfunction. Supposing you have programmed cell death protein 1 PD1, it is expressed by your CD4, CD8B lymphocytes, dendritic cells, monocytes, mast cells and Langerhans cells. It binds to its PDL1 and PDL2 ligand. Where are this ligand expressing cells? It has it in the antigen presenting cells, and it is there in your CD4 T cells, non-lymphoid cells and some tumor cells. Very important here is again your T lymphocytes associated protein 4, CTLA4. This also is very important, which is like it is expressed by your CD4 activated or even by exhausted Tregs, CD8 tumors, and it has this ligand at CD18 and CD86, and it is like this. These two ligands are expressed by your antigen presenting cells. Then the lymphocytes, this is a very brief examples I have kept it here only for our reference. For your CD4 expressing cells and the CD8 and the natural killer cells express this LAG3, and the ligands are your MHC class 2 and the L-sectin, and the ligand expressing cells are your antigen presenting cells and then some tumor. Here we have, now we know we have totally established what are immune checkpoints. Okay, how can we? So, if you target this cell membrane receptors, it is possible with such monoclonal antibodies, yes, and this can mimic or block the effector of a receptor or a ligand, and thereby they can enhance your immune response. Here you can see that you have the ligands, that is like your antigen presenting cells like BH7, like your PD1, PDL1, L2, CD60, CD66. You have the blocking antibodies, we will be discussing about them in detail, and then you have your T cell which will have the receptor, inhibitory receptor C. Very fantastic, your antigen presenting cells and your T cells. And here also you have some more antibodies and the ligand directed antibodies are also there. This recognition of this complex, it totally occurs via a T cell receptor. However, your T cell typically requires an additional signal, just not like by recognition of this, definitely an addition. This activation of this T cells is further regulated by the balance of your T cell of the inhibitor, that is your checkpoint, and the co-stimulatory factor, which are critical in healthy subjects to prevent autoimmunity and to protect normal tissues when the immune system is activated against a pathogen. Many immune checkpoints or co-stimulatory molecules such as these, on the APCs and receptor, they have been very much identified, and several people, several groups have worked on this. This if you have a monoclonal antibodies where you can block any of this, and then definitely yes, there is a better immune response. This is a typical mechanism of action. Again, here you have the tumor cells, and then you have the PD-1. Then this is the immunatory system. You have the reinvigoration of your T cells after inhibition of the interaction. You have supposing you have the reverse signaling via PD-L1 favors tumor growth and anti-apoptotic process. This PD-L1, it will promote the growth of the tumor cells. With the antigen presenting cells, the stimulatory such as your CD80, CD28, 86 and the CTL4, promoting CD28 over CTAL4 signaling after CTLA4 blocking. You have even the Tregs, your regulatory T cells, where after you deplete them by your anti-CTLA monoclonal antibodies. This is the mechanism of an antibody action of antagonist. You have the cytotoxic T lymphocyte associated protein 4 CTL4 inhibitors. CTL4 was first described by Leach et al. In 1996 as a receptor on T cells, where it acts as a physiological brake on the T cell activation. It competes with your CD28 receptor, stimulatory receptor on the T cell. Both bind with your CD80 and CD86 ligands, also known as B71 and B72 respectively, and these are which are seen on antigen presenting cells, but CTLA4 has a very high affinity for these two ligands than originally your CD28 has. If you block CTLA4 B7 interaction, it favors B28 B7 interactions. As a result, there is more increased in production of your T cells, then increased T cell survival, and further leading to activation of your T effector cells and increased diversity T cell response on tumor. This is ipilimumab. This is the mechanism of action. This is a drug, and then T cell activation requires stimulation through both the T cell receptors and your CD28. Here you can see binding of your B7 family proteins here, very fantastic, right? You have a T cell, then you have antigen presenting. Supposing the binding of this particular B7 family proteins to CTLA4, it inhibits your T cell function. CTLA4, therefore your T cell function is totally inhibited. Now here CTLA4 expression increases in parallel with your with T cell receptor stimulation, thereby serving as a brake on T cell response. Anti-CTLA4 antibodies here, as you see, such as your ipilimumab, these are monoclonal antibodies. They block your here can you see here how is this mechanism here and how is this? They will be blocking your CTLA4 binding to your B7. Isn't it fantastic? You have your co-signaling pathway which is totally being targeted here, and they prevent your inhibition of the T cell function. What will happen here? Here the effective function is your T cell is inhibition, in here your T cell remain active. This is a very path breaking research for ipilimumab, and there are several clinical trials for both melanoma, non-squamous cell lung carcinoma, which is improving the patient response rate. Here again we are coming again to one more immunotherapy, very famous molecule, programmed death 1 or programmed cell death 1. This people at Kyoto University, they discovered the gene which encodes the PD1 and the PD1 receptor, also known as your CD279, and which is a transmembrane protein receptor which is not detectable on resting T cells but is highly expressed by your activated T cells. Please keep in mind. Two ligands have been identified here for your PD1, such as your PDL1 and your PDL2. by these different groups, and they developed human monoclonal, first initially they developed human antibodies against PD-L1 and its ligand. They found that anti-tumor effects in mice are also were observed with these surrogate antibodies to mouse PD-1 and PD-L1. You have a very famous nivolumab, which is very fully human monoclonal IgG4 antibody, which binds very specifically to the PD-1 receptor, and it is very much used for your clinical development for not only for lung cancer but for even the breast cancer. So, nivolumab was also able to block the interaction of PD-1 with both its ligands. This is again another path-breaking immunotherapy for cancer. This is a very active clinical trial. It is very well documented. Both PD-1 and CTL4 pathways have non-reductant roles. Immune checkpoint inhibitors like your antibodies, they can attach to this particular to their co-inhibitory receptors, thereby reactivating your immune response against tumor cells. The different groups of immune checkpoint inhibitors, ICIs, so they include your nivolumab, your pembrolizumab, and the cemiplimab, and PDL1 inhibitors, very important, and they are very well successful in many clinical trials. Atezolizumab, durvalumab, and evolumab, and CTLA4 inhibitor. That is what we have seen, how nicely we have seen, right, in that diagram. These are all have been approved by the US FDA for the treatment of various types of cancer. As I mentioned, this is very highly selective. This binding of your T cell receptor and your MHC, it will activate the adaptive immune response. The binding of your PD-1 and PD-L1 can prevent your signaling transaction of the T cells to inhibit the immune response, whereas your anti-PD-1 antibody, here you can see this particular antibodies here, this is all this antibodies here. They can reverse the inhibition on the of this particular interaction. Therefore, please note here, cancer cells, how do they escape immune response? By overexpressing PD-L1. This immune system is activated not only by diseases, whereas PD-L1 inhibits the immune system by preventing foreign antigen specific T cells from accumulating and reducing antigen specific CD8 T cell proliferation. Please note here, increase in PD-L1, which is called which is expressed by this particular cells, it will inhibit the immune system by preventing foreign antigen specific T cells from accumulating and reducing antigen specific CD8 positive T cell proliferation. This again is a very fantastic diagram which talks about, how the anti-tumor immune response via PD-1 blockade by a nivolumab is achieved here. Very fantastic figure like the earlier one. For example, you have the recognition of tumor by T cells through the MHC antigen interactions, which mediates your IFN gamma release and PD-L1 and PD-L2 upregulation on. This is a very first thing what it happens. As we have seen in the previous slides, you have your upregulation. Your PD-L1 interaction, it mediates inhibition of T cell anti-tumor immune response. Then here you have the priming and activation of T cells through your MHC antigen and CD28B7 interactions with your antigen presenting cells. See here how does it happen here. This also it inhibits your T cell activation. You have the T cell anti-tumor response is inhibition and your T cell activation is inhibited. Then you have the recognition of tumor by T cells through MHC antigens, and which mediates your IFN gamma results and PD-L1 and PD-L2 upregulation on your tumor here. Here, can you see this? How even it happens here also. What happens when nivolumab happens? There is blockade of PD-1 and your PD-L2, which results in reactivation of your anti-tumor response. So, can you see here how this blockade did? See here, this is completely happen. This is completely this whole it binding happens, but whereas here once there is a blockage, what will happen? Your complete reactivation of your anti-tumor immune response. This strategy behind your immune checkpoint blockade is to reduce the inhibitory signal and restore the patient's natural tumor specific T cell mediated response, which is achieved by this particular monoclonal antibody. Very important function and very successful clinical, many trials have been enrolled for this particular. Your this particular drug, this will result clause in the blockade of your PD1 and or PDL1 and L2 and reactivation of a anti-tumor response. After, since the advent of this particular drug, there has been an durable response and there has been an increase in overall 5-year survival rates for 20% of these patients who receive this particular drugs. This is an example of the different drugs, you know, and these are the different pharma names and these are the different manufacturers, and there have been very well successful. You even have it in your metastatic cutaneous squamous cell carcinoma, then for even, and this is not only used per se in the monoclonal, but along with your, as I mentioned, for in the surgery or with other radiation procedure. This is your different landscape for over the time from over the 2017 and 2018, where different clinical trial for PD1, PDL1 immune checkpoint inhibitor drugs. These are all the different trials which they have used, and many are still in progress, and each bubble here, the number here, it represents the number of clinical trials. For example, they are going to use this particular drugs in conjugation with the other particular targeting drugs, pathway targeting drugs, like, and even like for example a chemotherapy and then your CTL4 or a radiotherapy and your CTL4 or your other pathway. Now coming to another term which we will introduce is your predictive biomarkers. Please just one thing, go back to your cancer detection methods where we totally discussed about what exactly are the biomarkers. Now we have shown that you have a ligand, then you have a receptor. Now can we check this PDL1 status before you proceed further for the treatment of the patient? Yes, it is possible. And then this you check it like as I said, this is your tumor microenvironment which represents heterogeneous milieu of cells. Before then since both immunocytes and your cytokines here, you know, they all play a very good role in mediating immunosurveillance and regulating tumor cells. This heterogeneous nature of your tumor will have a very fantastic effect in influencing the efficacy of this immunotherapy. These markers which are measured using, there are many well validated in vitro use assays, even like your FISH and then your IHC, and then you even have your NGS, which we have discussed before. They can help in maybe if you can go for your PDL1 status, what is the tumor mutation burden. They can help in enrichment of the patient population for clinical trials, and then you can stratify the patient easy PDL1 positive, PDL2 positive. Now how do we go about with the treatment strategies? So another immunotherapy frontier is your cancer vaccines. So they are there are like it has been a subject of very well review, and many of them have questioned what exactly the antigen can be, which can be introduced. Do you want to introduce a whole tumor or tumor cells, proteins, peptide based vaccines? Then you have your RNA, then you have your DNA, and then your dendritic cells, proteins. We will not be talking into all, but very briefly current vaccines we will see how they are classified. Now, what is known of based on what is known of a tumor specific immunogenic antigen, or which patient's tumor express those antigens, how the antigens become co-localized with professional antigen presenting cells. First thing, vaccines can can incorporate either predefined or anonymous antigens, which is like we really know what exactly this particular antigen. They require predefined vaccines, required your identification of antigens either by a tumor biopsy and computational analysis. Is it a personalized vaccine? Yes. So, are these antigens defined before treatment? Sometimes if you have no, this algorithm says, then are these antigens, are they loaded into your antigen presenting cells outside the body? If they are no, is it through the in situ or through the ex vivo? So, tumor cells, lysates containing antigens of the excited tissues are loaded into your autologous antigen presented the cells, and they are injected back into the patient. Are antigens expressed in many patients with the same tumor type? This is very is it like, isn't it a fantastic when the same tumor many patients express the same antigens, then personalized, it is not definitely personalized, but if it's not, but cancer specific mutations, they produce novel epitopes or neoepitopes or neoantigens, what we discussed, and these are only specifically expressed only on specific cancer cells from select patients, or maybe, and sometimes this is a very broad group, this is shared, yes, predefined antigens. So, cancer specific mutations produce mutagens, that antigens that are highly expressed in the same cancer type, but in many patients. Anonymous antigen vaccines can co-localize antigens with APCs in the laboratory or directly at the tumors. This is the mechanism of your action. For example, mechanism of cancer vaccine in vivo. After you have the tumor antigens which migrate to the antibody in various different forms, they are phagocytized and then intracellularly expressed and efficiently processed by your APCs, your typical APCs. Then the major histocompatibility complexes of your dendritic cells, they present antigens to their surface and employ MHC complex antigen specific T cells by binding. This MHC complex will activate your T cells, antigen specific T cells by binding to your T cell receptors, and on the surface of the T cells, therefore, safely, persistently and specifically destroying only tumor cells and inhibiting tumor growth. This is like a very soluble antigen is presented, your T cell priming and activation happens, your CD4, CD8 mechanism, then your T cells will migrate and they will infiltrate your tumor, recognized and killing your specific antigen cancer cells, and again further more antigens is released from your tumors. The first step is only a simple or small amount, where this itself will cause an exocrine release of several more antigens, which will in turn cause T cell priming and activation, thereby reducing thereby trying to increasing the T cell response. So, first thing what is very important here is your T cell. Again, as we mentioned about your tumor specific antigens or tumor associated antigens, you need to have an optimal tumor antigen selection in cancer vaccine. You have the categorization of your 4 vaccine types, or what is known as the predefined versus anonymous, and then where patients tumors express those TIAs, shared versus personalized, and how antigen presenting cells they encounter and load TAs ex vivo versus your in situ. Here your shared antigen vaccines target antigens commonly present expressed across multiple cancer vaccines, targeting specific mutations in your individual tumor, making them off shelf and low cost immunotherapies. This is how a distant tumor is totally targeted by your specific T cells. This is like tumor antigens, as I mentioned, like you even based on the you have the shared antigens and the public antigens, which contain the hotspot mutations. What are hotspot mutations? Which is almost most very frequently mutations by a and then by a relatively low common they target your tumor associated antigens and tumor specific antigens. And, as I said before, tumor associated antigens are present even in your normal tissues. Of course, they are present in low quantities and they are not overexpressed, unlike in cancer. But whereas your TSAs is directly produced by your cancer, by your tumor cells. Your melanoma associated antigen such a major which is normally expressed and overexpressed in testis and melanoma, whereas your HPV associated the cervical and oropharyngeal cancers have high expression of your EC6 and your E7 proteins of high risk HRSPB. Therefore, the shared TAAs in the patients with cancer make it a promising off the shelf immunotherapy of option. So, you have simply something called as personalized. See, isn't it like, if you recollect, we are talking about personalized therapy, how an individual will have a different receptors and a different cancer profiling or stratification of the particular cancer or a tumor is done. The same way, if that is transformed, isn't it fantastic to talk about a personalized vaccine? Therefore, your shared TAs in patients make a promising off the shelf immunotherapy option, whereas your personalized cancer vaccines, they have gained the spotlight due to modern high throughput. This is only because of your high throughput genome sequencing, where exactly you are able to totally pinpoint what exactly the tumor mutation is, and then and then you are able to give a deeper understanding to your neoantigen production, thereby informing can you have a personalized vaccine for that particular tumor type. These are the different history and the key time points. First thing, the concept of your immunotherapy, which was we have already discussed about this particular which was inspired by the observations of your spontaneous cancer regressions after infections like your colitoxins in 1893. Then Lloyd discovered that BCG prolongs the survival of mice with cancer. Then in 1984, Hover's pioneer use of your vaccine, which was made from tumor cell lysates with increased responses of autologous tumor cells in patients with colorectal cancer. Then following by, you have the TAA or the MAGE1 was identified in the clinical trials of TAA based clinical vaccine were conducted since then. Then this was Melasine was the first approved to be market tumor lysate vaccine against your melanoma. And then you have the peptide based vaccine at 2000, very important breakthrough, which these peptide vaccines, they served as a strategy to stimulate immune response against specific antigens. And then we even had the FDA fully approved dendritic cell vaccine, that is your Supilizumab T for metastatic castration resistant prostate cancer. And then you have the several phase 3 clinical trials of therapeutic cancer vaccines which are targeting TAE, which, and here again of course there are quite a few failures here. And then your neoantigens were discovered here. Then the first human test of your conceptualized mRNA cancer vaccine was tested in 2017. Then coming to 2023, they have focused on not only on vaccine formulations, but also more effective delivery methods, and then combination of various therapies for various combination along with other therapies also. The phase 3 clinical trial of mRNA and personalized cancer vaccine, then along with pembrolizumab is conducted in patients with post-operative melanoma. This is by far how the history and how the different milestones, how the cancer vaccines have evolved over the time period. So, now what is the future more roadmap for this particular immune checkpoint inhibitors do we need to have? strategies to select combinations for testing. As I said before, the different drugs, we have so many, we just discussed only 2 drugs, but what about if you have many more? Yes, we have many more drugs. How do you have a strategy for selecting? Yes, this is very, very important. As I said, now again we are stressing, not one size fits for all. So, not a single combination would be taken up for treatment for a single cancer. Role of importance of biomarker. This is very, very, very important because these biomarkers are, as we said, like PDL1 staining, or they will predict how the patient's outcome, or what is the response to that particular combination of this particular inhibitors. Can we improve? How do we have strategies to improve on your clinical trial designs? Yes, it is very, very important to have different clinical trials where several immune checkpoint inhibitors are used. And then what are the regulatory challenges and opportunities? Yes, the challenge is lots of cytotoxicity, as like with other drugs, but not it's minimum. And how does this whole data has to be very authentic. Suppose several places are connecting clinical trials, yes, they should be putting up, and then that the real world evidence is available. This is the cartoons which predicts how the T cells will fight the cancer. Then you have exhausted T cells after the fight, and then you have your reinvigorated T cells, which again, like with your immunotherapy or whatever you have given. And then we haven't discussed here, but if you have this IL-10 substitutions along with this particular inhibitory molecule, monoclonal antibodies, yes, there is an improvement in the immune response. This is by far the sessions including your introduction to immune therapy, where we have introduced to the different terminologies and the different drugs, and then what exactly how do this mechanism of action, this particular 2 drugs we have seen for 2 different and pathways, different molecules such as your CTLA4 and then your PDL1 and the PD1. Thank you.